Now, let us look at some other miscellaneous targets. So, we have looked at earlier what transport proteins are, and so these are proteins which are sort of help in transporting important metabolites. So, for example, we can look at amino acid transporters or transporters of neurotransmitters and so on. Okay. So, if we can there are drugs which can bind to these transport proteins and prevent the normal functioning of the protein. So, some examples are cocaine and some tricyclic antidepressants, and these go and prevent neurotransmitters from getting back into the cell. So, once it prevents this, now because it is present in the synaptic area, it results in increased level of neurotransmitter activity. Okay. So, next example is inhibitors of tubulin polymerization. We have already looked at previously that polymerization of tubulin is an important process during cell division. So, an example of an inhibitor of polymerization. So, what will happen is that if you inhibit the polymerization, then you are able to prevent the cell from dividing, and this is extremely useful in the treatment of cancer, for example. So, colchicine, whose structure is shown here, is one such example of a inhibitor of tubulin polymerization, and this compound is actually used in the treatment of gout. So, where there is quite a bit of swelling in certain areas of the body. There are other natural products and natural product like molecules which are also known to have tubulin polymerization activity. An example is vincristin, vinblastin and so on, and these are alkaloids which are extracted from natural sources, and these bind to tubulin and prevent the polymerization. And these complex molecules have very useful anti-cancer activity. Okay. And some of the analogues of these compounds are being examined or being studied as potential anti-cancer drugs. The counter view or the opposite example is in tubulin depolymerization. So, tubulin depolymerization also needs to occur, because this is a process by which the normal the cell functions normally, and so inhibition of depolymerization also becomes an important way to inhibit cancers. So, the natural product Taxol, paclitaxel, which is commercially known as Taxol, and the semi-synthetic analog. So, semi-synthetic analog means something that has a structure which is similar to it, but some of it is actually synthesized, and these are known to inhibit depolymerization. Okay. And there are a range of other natural products which have also been found to prevent the polymerization of microtubules and so on. Now, the other way in which one can design new drugs, or one can look at drug targets, is to inhibit biosynthesis. So, we have already looked at in detail the biosynthesis of proteins. So, proteins are synthesized by by using the machinery of RNA, and RNA has this amino acyl tRNA, which has which is shown here, which is then bound to the amino acid over here as shown here, and this is the charged or the armed tRNA, and then it goes and binds in the ribosome, and once it binds, then it is going to transfer this amino acid to the growing peptide chain. So, puromycin, whose structure is shown here, is a very good example of a inhibitor of this process. Okay. So, what happens is that this actually mimics the terminus of the amino acyl tRNA molecule. So, you see here the structure, here you have the adenosine part remains the same, the sugar part has very similar structure as you can see here, and the and there is a amino acid here, but the amino acid is now bound to an amide instead of a ester. So, once it goes and binds to the ribosome, it acts as a way in which you can inhibit the biosynthesis. So, let us look at the example here. So, what puromycin does is that it comes here and it can react with the growing peptide chain, and once puromycin is bound here, there is no further reaction possible, and so it dissociates and it prevents the protein from growing. So, once the protein so it has the amino group required for the transfer reaction, and so the peptide chain is transferred from tRNA in the P binding site to puromycin in the A binding site, and then puromycin departs and carrying the protein, but the protein now is stunted. Okay, so let us say it is supposed to have 250 amino acids, but let us say this process occurs at around 85, 87, then it stops at that process, and then the entire protein is not synthesized. So, in this manner, what happens is that puromycin can inhibit the biosynthesis of proteins, and because the protein is now stunted, the function of the protein is not the same, and once you have the function of protein inhibited, the cell can it can be fatal for the cell. So, puromycin is a very important example of drug that inhibits biosynthesis. Now, in a cell, we have many components. So, we looked at proteins which are made up of amino acids, and these proteins can be part of enzymes, or it can be part of receptors and so on. And you also have importantly DNA, and which is nucleic acids, and we have already looked at how to target nucleic acids. The third major component inside a cell is lipids. So, lipids are basically hydrophobic molecules. They are typically long chain molecules which have a lot of carbons in it, or they can also be cyclic molecules such as cholesterol, but they are extremely hydrophobic in nature. And we have already looked at previously that lipids are important components of the cell membrane. Okay. And so, if lipids are considered as drug targets, then what would happen is that they would disrupt the lipid structure of the cell membrane, and once this is disrupted, the protective layer around the cell is now become less efficient, and the cell can rupture and die. Okay. So, the first example that we look at is the antifungal agent amphotericin B. So, this is known to interact with lipids and sterols on the fungal cell membrane, and what it does is it helps in building tunnels. So, we look at how the process happens, but once this tunnel is made, then the contents of the cell are now going to be drained away. There is going to be imbalance inside the cell, and the cell dies. There are 4 major regions in amphotericin B, and so the first one that we look at is the hydrophobic region, which is imparted by this long chain olefin rich lipid region. And there is also a hydrophilic region here. You have a number of hydroxyl groups over here, and then there are 2 hydrophilic regions. One is composed of an amino sugar, which is shown here, and the other one is a regular sugar with a carboxylic acid on it. So, essentially one half of the structure is made of hydrophobic regions, and it contains a number of double bonds, and the other half contains hydroxyl groups and is actually hydrophilic. So, this makes the molecule very unique. Now, because of this very unique structure, what happens is that several amphotericin molecules can actually cluster together. And the way they cluster is that you will have the hydrophobic regions actually interacting with other hydrophobic regions, and the hydrophilic regions will interact with the hydrophilic regions. So, as we already know that in a cell membrane, the area is mainly made of lipids, and lipids are the center of the internal part of the membrane is quite hydrophobic, and the external part of the membrane is quite hydrophilic. So, once amphotericin B, the hydrophobic part now interacts with the membrane, then you can think about the hydrophobic regions actually coming close to each other and forming favorable interactions. And this exposes the hydrophilic regions to the other side. So, you can imagine a number of such molecules which are going to sort of self assemble in this manner, and in the process what happens is that it can form some sort of a tunnel, and this tunnel resulting from this cluster is lined with hydroxyl groups. And so, what can happen is that it allows for the polar contents in the cell to drain away. And so, once this is drained away, it causes ionic imbalance and which can lead to cell death. There are some recent studies which have shown that amphotericin B actually forms a very important favorable hydrogen bonding interaction with ergosterol. Ergosterol is the fungal equivalent of cholesterol, and so this is the structure of ergosterol over here, and it is quite hydrophobic in nature. But there is a hydroxyl group at the terminus at one of the ends which can interact favorably with amphotericin B. There are other peptides which are also known to form these kinds of channels or pores. So, gramicidin A, which is whose primary structure is shown here, is a 15 amino acid containing peptide, and what it can do is that because it contains both hydrophobic as well as hydrophilic regions in its peptide structure, it can interact favorably with the lipid membranes. So, what can happen is that you have the hydrophobic regions of the peptide which are going to form an alpha helix like structure, and they are going to interact with the lipids, and you can imagine that these are going to be stacked up, and once it stacks up, it allows for ions to flow through it because the hydrophilic regions are going to be inside. So, again this is a process by which ions can then escape from the cell, and it leads to ionic imbalance and cell death. So, gramicidin A can also be viewed as an escape tunnel through the cell membrane. So, once this ionic imbalance occurs, the cell dies. One molecule of gramicidin itself would not be long enough to traverse the membrane, and so it appears that there will be two gramicidin helices align themselves from end to end to achieve the length required. So, there is another class of polypeptides which is called megainins, and this is again a helix, and it contains 23 residue polypeptides, and these are antibiotics, and they form helical structures which disrupt the permeability of cell membranes. So, here is an example of the cell membrane. So, once it comes, it binds to this helix binds to the upper portion of the lipid membrane, and then it actually aligns itself through process of self assembly, and it forms a pore. So, this cyclic peptide is shown by this cylinder over here, and this cylinder can actually stack itself, right, and it forms what is known as a nanotube, and this nanotube can actually disrupt cell membranes. Okay. And once it is formed, the nanotube would allow molecules to leach out from the cell and cause cell death. If you look at closely at this nanotube, these structures have 6 to 8 alternating D and L amino acids, okay, such that the amino acids, the amide groups are perpendicular to the plane of the cyclic structure. So, you have the amide group here and amide group over here, and these are actually going to be perpendicular to the chain of the structure. And once this forms a hydrogen bonded network, then it can form multiple of these cyclic peptides can align themselves one on top of the other. And the side chains do not interfere with the stacking process, and the side chains are actually exposed to the outside. And each cyclic peptide forms hydrogen bonds to the cyclic peptide above and below it, and once this forms, then it can form a nice self assembled stack which can form a sort of a nanotube. So, what it allows us to do is, if we can modify the side chains, then we could think about imparting selectivity in vitro for bacterial cells versus RBCs. So, otherwise what happens is that these cyclic peptides are known to be toxic to red blood cells as well, and they can cause what is known as hemolysis. So, for example, you can include the basic amino acid lysine, which can be useful for such selectivity. Lysine has a primary amino group which we have already looked at earlier, and this can allow for protonation, and once it gains a positive charge, it can make it ionic. So, this entire process enables the structures to target bacterial membranes, because bacterial membranes tend to have a negative charge on the surface, and a number of animal studies have been carried out, and these molecules have been found to be having very promising activity. and further work needs to be done on this at this point, but they become good starting points for further drug discovery. The next class of molecules are known as ion carriers. So, the example that we are going to look at is valinomycin. So, valinomycin is again a cyclic structure as shown here, and it contains 3 molecules of L-valine and 3 molecules of D-valine, and it also contains lactic acid and hydroxy isovalerate. So, this is the structure of lactic acid as lactate as shown here, and this is the structure of hydroxy isovalerate. So, together this forms a cyclic structure, and these 4 components are, there is an alternating sequence of ester and amide linkages around this cyclic structure. And so, this is achieved because we have an important molecule such as lactic acid or hydroxy isovaleric acid between the subunits of each of the 6 valine units. So, as shown here, so you can see here the ester linkages which are present between the neighboring amide units. So, what happens is that this is actually clearly this structure is favored because the hydrophobic chains can interact via van der Waals interactions. So, you have here this hydrophobic chains which can interact with each other, and these can actually interact with the fatty lipid interior of the cell membrane. So, we have already looked at, so the cell membrane has the inner part of the cell membrane actually is hydrophobic, whereas the hydrophilic groups are clustered together in the center as shown here. So, what it can do is that it provides a hydrophilic environment inside the molecule, whereas the hydrophobic environment is outside, and this allows for the structure to actually go and bind to lipids. And this resembles what is known as a donut. Okay, so, donut is basically a food item which has a structure such as this. So, where there is a hole in the center and it has a sweet exterior. So, the hydrophilic center is large enough to accommodate an ion, okay, and it is found that a naked potassium ion can actually pass through this. So, what I mean by naked potassium ion is that it does not have water molecules around it. As we know, any ion that is present inside the cell or inside solution is going to have, let us say M plus, it is going to have water molecules around it. So, this is called, you have interactions over here, and you have a number of water molecules surrounding the molecule. So, this is actually called hydration, and hydration is important because it stabilizes the positive charge on the species. However, if the metal ion is actually very well hydrated, then what it does is that it effectively increases the size of the metal ion, and because of hydration we have to deal with what is known as a hydrated ionic radius. So, hydrated ionic radius is nothing but the radius of which is or the size of the molecule, or like we assume that it is a sphere, and this sphere which contains the metal ion as well as the water molecules, and together they behave like a single unit. Okay. So, valinomycin actually collects a potassium ion, not a hydrated potassium ion, but a naked potassium ion from the inner surface of the membrane, and then it carries it across the membrane and takes it to the outside part. So, this is the mechanism. So, potassium ion becomes associated with the with valinomycin, and valinomycin, as we know, has a very hydrophobic exterior, and then once it binds, then it goes in, and then the potassium ion is delivered outside the cell, and then valinomycin can do this again. So, we just looked at example of potassium ion, and we have data to show that valinomycin is actually specific for potassium ions, and it does not work with sodium ions. Is it that sodium ions are too small? We already know that sodium ions and potassium, sodium is has a smaller radius compared to potassium ion, but what happens is that sodium ion actually has a larger hydrated ionic radius compared to potassium. So, as such, sodium ion is too big, the hydrated sodium ion is too big for the central cavity of valinomycin. So, and sodium ions do not lose their surrounding water as easily as potassium ion. So, potassium ion being larger has a greater tendency to lose the water molecules and become naked, whereas sodium ion does not do this. So, together this imparts selectivity towards potassium ions over sodium ions. The next set of molecules which we are going to look at are nigericin and related molecules, whose structure is shown here, and these have been used in animal medicine, that is veterinary medicine, as antibacterials, and they function quite similarly to valinomycin. So, we have looked at again proteins, we have looked at nucleic acids, and we just covered the topics of lipids. The next major class of molecules that are present inside the cell are carbohydrates. So, the term glycomics is used to describe the study of carbohydrates, and these studies are carried out using carbohydrates as drugs or drug targets. And just to recap, carbohydrates are polyhydroxy structures which have the general formula CN, H2N, ON, and these are simple examples of simple carbohydrates include glucose, fructose, ribose and so on, and we have already looked at ribose as an important component of nucleic acid, and glucose is important from the energetic standpoint. Now, the structure of fructose and ribose is shown here. Now, these are known as monosaccharides, okay, and these are essentially monomers, and these can combine and form oligomers or polymers. And because sugars have multiple hydroxyl groups on it, the combination can be fairly complex. So, an example here is with glucose, you have multiple hydroxyl groups which can interact with the next molecule, and so you can have O glucose combining two glucose molecules combining through one of these hydroxyl groups and gives you one structure, and you can see here another combination here, a third combination, fourth combination and so on. So, these molecules can combine to form incredibly complex structures, and there are a number of natural polymers such as glycogen, cellulose or starch, which are basically combinations of these sugars. Now, for carbohydrates to be considered as drug targets, the reason why they are interesting is because they have important roles to play in various cellular processes. Very key role that carbohydrates play is in cell recognition. So, the surface of the cell has a number of sugars which enable recognition of like cells. For example, humans have present certain sugars on the surface which help the human cells to recognize itself, and it also helps in understanding what are the sort of pathogenic cells there and get rid of it. So, they are very crucial for cell recognition, also for cell regulation and cell growth. Now, bacteria and viruses have to recognize host cells before they can infect them, and these carbohydrate molecules are involved in the cell recognition process. So, designing drugs that bind to these carbohydrates, the hypothesis is that they may block these interactions of bacteria and viruses and prevent invasion of host cells. So, therefore, carbohydrates are important drug targets. Lastly, we are going to look at antigens and antibodies. So, we are not going to spend a whole lot of time on this, but these are basically tags that help with cell recognition, and antigens and antibodies help us understand if a cell is the same or it is a foreign cell. So, bacteria have their own cell recognition molecules which are different from our own. So, we have a very complicated set of antigen antibody system which can help with immune response. So, we are not like I said, we are not going to spend a huge amount of time on this, but an immune response is triggered when there is a recognition that there is a foreign body that is, or an invader that has come. And once this immune response is activated, it destroys the invading species. Antibodies are Y-shaped molecules that are made up of two heavy and light chains. Okay. So, here is the structure of that, and the N terminus of these chains has a highly variable region of amino acids, and these can differ from antibody to antibody. it is this region which recognizes the antigen. Okay. Once an antigen is recognized, the antibody binds to it and recruits the body's immune response to destroy the foreign cell. So, the body does not normally produce antibodies against its own cells, so we are typically safe from attack. However, when there is an invading cell such as a bacterium or a virus, then these antibodies will be produced, and these are going to help with getting rid of this of the foreigner. Now, there are also cases where people have organ transplants, where an organ from one individual is transplanted to the other individual. Now, since the receiver of this transplant is going to recognize this new organ as something that is different from itself, there could be rejection of the transplant. So, what one way to address this problem is to use a close match, which is basically genetically very close, so that the recognition may be similar, or the recognition is not a major problem. However, immunosuppressant drugs are also used, and these reduce the rates of rejection of transplants. Antibodies can be either used by themselves to mark cancer cells or as a means of delivering anti-cancer drugs. So, the new field of antibody drug conjugates has emerged in the past decade or so, wherein the antibodies are used because of their highly specific nature. They are used to mark cancer cells, and along with this you can deliver a drug, and that will help in highly selective killing of cancers.